So today's lesson is going to be about biomolecules, which are polymers, carbohydrates, proteins, lipids, and nucleic acids. After this lesson, you'll be able to explain the building block idea of monomers and polymers, relate the function of carbohydrates, proteins, nucleic acids, and lipids to the structure and function of a cell, and then distinguish between the structure and functions of carbohydrates, proteins, nucleic acids, and lipids. So polymers, these are macromolecules, and they are a large molecule comprised of smaller molecules that are joined together. So macro means large, and molecule means molecule, obviously. So macro is your first Latin prefix, and it means big or large. A monomer is a small subunit that composes a polymer. So mono means one. So mono, one subunit. A dimer, so di, means two monomers bound together. Polymer is a large molecule composed of similar subunits joined by a covalent bond. So poly means many. So here we can see a monomer. We have one subunit, a dimer with two, and polymers with multiple. So carbohydrates, these are types of food like bread, and they contain carbohydrates, most commonly known as sugar. So a carbohydrate is a molecule comprised of a carbon, a hydrogen, and an oxygen atom, and it is close to a ratio 1 to 2 to 1. Often these form ring-shaped structures. So we're going to see different ring-shaped structures. The most common one that we talk about is glucose, and it's C or carbon 6, hydrogen 12, oxygen 6. So we can see that 1 to 2 to 1 ratio. So we can find in single sugars containing 1 or 2 sugar rings, or starches containing 3 or more sugar rings. A monosaccharide is the simplest sugar. So mono means one, saccharide means sugar. So we have one sugar. It can exist on its own as a monomer of or in a carbohydrate polymer. So an example of this would be glucose. Oops. So disaccharides is a substance containing two monosaccharides bound together. So examples of this would be maltose with 2 glucose, sucrose, which is our sugar, like table sugar, which is glucose and fructose, or lactose, which we can see in milk, which is glucose and galactose. Right? Glucose and fructose make sucrose, which is typically our granulated table sugar that we eat and put in things. We can see them here. They join together with this oxygen in something called a dehydration synthesis. So you can remove water to form these bonds. So polysaccharides is a group of several monosaccharides bonded together through a dehydration synthesis. So once again, you can see here, we're pulling out water to bind these together. We can find these things in starches like potatoes, in our liver glucagon, which controls our blood sugar level. We can see them in cotton, and then we can see them in cellulose, chitin, which is the outside exterior of most invertebrates. So when you step on a cockroach and you hear that crunch, you are crunching their exoskeleton that is typically made out of chitin, which is that exoskeleton shell, right? That's crunch or chitin. So functions relating to cell structures, right? Carbohydrates provide energy sources, right? We use glucose to make energy in something called a glycolysis reaction. We break the carbon-hydrogen bonds to release that energy. We can once again see that in glycolysis, where we take one glucose molecule and make several different molecules of energy called ATP. We can also see them used in structures to stabilize. So rigid polysaccharides can be found in the cell membrane to make sure that they are straight and tight, so nothing can come in or go out. We can see these for protection and structural integrity. They can also be used for communication. So we've talked about carbohydrates, and we're going to move on to proteins. Proteins are a polymer comprised of amino acids. So amino acids are the monomer of protein. So we can put amino acids together to make that polysaccharides. They consist of a central carbon, a hydrogen atom, an amino group, and a carboxyl group with a variable R group. So if we change the R group, right, if we change the structure, we're going to change the function. So there are 20 different R groups. That means there are 20 different types of amino acids. Like I said, if we change that R group, we change the structure, which changes the function because structure dictates function. I want you to draw this right here in your notes. So I want the amino group, the carboxyl group, your hydrogen, and your R or your variable group down at the bottom. So a polypeptide is a group of several amino acids bound together. And a protein can be comprised of one polypeptide chain or several chains folded together in and of themselves. So proteins can be specific, and we can do this by changing the R group, and that will change the protein, right? If we change the structure, we change the function, or we can change the sequence of amino acids. Once again, we're changing the structure, so the sequence of amino acids, and that changes the protein. Each protein has a specific shape, giving it a specific function, right? Structure has a specific structure, and it gives it a specific function. So structure dictates function. So protein structure, there are 4 different levels of protein structure. Our primary structure is just the sequence of amino acids. So it's a long straight chain of amino acids. There's no folding in on itself. Then our secondary structure, hydrogen bonds begin to form in the polypeptide backbone. So they start twisting in of themselves. Secondary structures have 2 shapes. They're called alpha helixes and beta sheets. And then our tertiary structures are interactions between the R groups. So those alpha helixes and beta sheets start folding in on themselves and connecting to other parts of the primary amino acid structure between those R groups. And then our quaternary structure are interactions between 2 different polypeptide chains. So proteins become functional after the tertiary, right? They become functional here. So these are functioning tertiary structures, and quaternary structures are functioning proteins. Not all proteins have to be quaternary structure, but they all have to be tertiary structured. So you can see that here, right? We have our amino acid chain. Then we've got our alpha helix right here and our beta sheets, and those start to interact, and then they interact with other polypeptide chains. So the most popular example of a quaternary structure is going to be our hemoglobin or red blood cells. Those are 4 polypeptide chains coming together to carry oxygen molecules around our body. So functions of proteins. We've got fibrous proteins, which are groups of long fiber-like proteins that form structural parts of cells and body tissues that hold us together. Then we've got chemical messengers like globular proteins that are comprised of different proteins and serve various functions. So we've got globule proteins in our mouth that produce our saliva. We can see them on our skin to produce our sweat. We can also see globular chief cells in our stomach to produce our hydrochloric acid to digest our food. So proteins do quite a bit in our body. They hold us together, right? You can see here, they make our skin tight and young looking. They hold our joints together and our muscles onto our bones. They also produce things like hydrochloric acid so we can digest food. They can taste our food. They can make us smell, all beautiful things. So proteins are awesome. Nucleic acids are polymers responsible for reproduction and genetic material in proteins. So we're going to talk a little bit more about nucleic acids in our next chapter, but they're super cool and awesome, and they make you who you are today. So nucleotides are the monomer of a nucleic acid. So you can put a bunch of nucleotides together and you're going to get this long chain to form a polymer, or our DNA and RNA. They are composed of a phosphate group and a 5-carbon sugar with a nitrogenous base. And then the nitrogenous bases are going to be adenine, guanine, cytosine, thymine, and uracil. So we've got 5 nitrogenous bases, and they have these really cool rules that we'll go over in our next chapter. But just know nucleic acids are awesome. So here we can see we've got our 5-carbon sugar, which is deoxyribose, our phosphate group, and here's our nitrogenous base, which is cytosine. So here are some differences in those polymers. So DNA and RNA are 2 polymers. DNA is usually double-stranded. Our sugar is going to be deoxyribose, and the bases that we are going to have are adenine, guanine, cytosine, and thymine. While in RNA, we typically have 1 strand. Our sugar is ribose, and the bases present are adenine, guanine, cytosine, and uracil. So here we have DNA. You can see it is double-stranded, and then our RNA is single-stranded. So structures versus function. So the structure, right, we have nitrogenous bases that protrude from the backbone to form rungs on a ladder. So if you've ever seen the little cartoon GIFs of DNA molecules, you can see the rungs in the middle and it kind of twists around them. Those rungs are nitrogenous bases. The information is stored actually on those nitrogenous bases. So the order that the nitrogenous bases go in is what makes the code or stores the information. And it's like binary for computers. Functions, the DNA holds information for protein synthesis and passes the information from one generation to another. So your mom and your dad each gave you part of your DNA, and when you have children, half of your DNA will be passed on to your child. And RNA takes the information from the DNA, it takes it out of the nucleus, and then goes and makes a protein. So lipids are a completely different type of macromolecule, and they're not comprised of specific polymers. Lipids we're going to see be fats, oils, steroids, and these guys are composed of carbon, hydrogen, and oxygen. They are not composed of a specific polymer, like I said, and they are typically hydrophobic or insoluble in water. So if you've ever poured oil in water, you know that they separate, and this is due to the presence of non-polar hydrocarbon chains. So those long chains make oil not like water. So oil and water don't mix. So fats and oils, we have triglycerides, and these are fats and oils consisting of 1 molecule of glycerol bound to 3 fatty acids. So it's a glycerol, which is a 3-carbon chain bound to a hydrogen atom and an alcohol group, and fatty acids, which are the long hydrocarbon chain, which is a carboxyl group on one end. So you can see here, right, glycerol is this one molecule with 3 carbons. And then we have fatty acids with all these carbons bound to hydrogens and that one carbon bound to oxygen on the end. Then we have saturated fats, which are butter and lard, and they're saturated because they are filled with hydrogen. They have so many hydrogens they can't bond with anything else. All carbon atoms are bound to either another carbon atom or a hydrogen atom, so they're saturated with hydrogens and they're solid at room temp. So, right, they're saturated, they've got all these carbons and all these hydrogens. Then unsaturated fats are typically going to be our vegetable oils, olive oils, avocado oils, things like that. And they are unsaturated because they don't have all those hydrogen bonds. Some of the carbons are double bound to other carbon atoms, giving them these kinks or bends in that fatty acid tail. These are going to be liquid at room temperature, right? Your olive oil, your avocado oil are both liquid at room temperature. So you can see them here, right? We've got those kinks in the tail. That's where our double bonds are. So lipids play a super important role in the composition of cells. They make up cell membranes, and they have these things called phospholipids. The cell membrane is comprised of millions of phospholipids. We have a hydrophilic head, which is a glycerol phosphate or charged molecule. And they love water. They want to be around water. They want to be saturated in water. They are hydro means water, philic means like. They like water. Hydrophobic tails, which are the 2 fatty acids, hydrophobic, hydro means water, phobic means hate, so it is horribly afraid of water, doesn't want anything to do with it. And these are 2 fatty acid tails. So hydrophilic means soluble in water or likes water. And we can see it here, right? So we have our hydrophilic head, and then we have our hydrophobic tail. And each of our hydrophobic tails has 1 saturated fatty acid and 1 unsaturated fatty acid. And this is really important when we talk about cell structure later on. I just want to point that out to you now. And then, right, so they're important component to cell membranes. I've already said there's millions of them in the outer membrane wall. And this forms the phospholipid bilayer, which the hydrophilic tail turns inwards towards each other. The hydrophobic tail turns inwards from each other. And the hydrophilic tails are on the outside of the membrane because they like water. So you can see them here, right? We have our hydrophilic heads up on the top and our hydrophobic tails on the inside, and then our water around it. So we don't really like water. We love water. Don't really like water, love water. So our last type of lipid that we are going to talk about is our steroids. And it's a type of lipid that consists of a backbone that has a 4-carbon ring backbone. And these are really important because they help maintain structure. So cholesterol is an important component of a cell membrane because it adds rigidity to the cell wall. We can also see steroids being messengers in the body. So structures is a large amount of carbon and hydrogen bonds. Their functions is to stabilize that cell membrane. If we didn't have that cholesterol, our cell membrane would be loosey-goosey and would separate and fall apart. We can also see steroids being chemical messengers. So we can see our cells send other cells steroids to say, hey, produce this, or hey, produce that. So here, right, we've got 4 rings, 2, 3, 4, and that is our steroid. Right, and then finally, here's a little chart, and I'm going to give this to you to put in your notebooks. So you can see the little differences between all of them. If you have any questions, please don't hesitate to reach out. I'm always around to answer questions.